trying to catch up on the parts that we have not covered yet for proteins. And then by next lecture, we'll be starting with nucleic acids and everything else that goes with it. And yes, I do record all my lectures. So here, Face ID is not recognizing me. Okay, so here I also do let's make a sound backup of my lectures because sometimes the PowerPoint acts in a strange fashion and I lose all the sound. And now I cannot find the other. Okay, got it. So, before I start, and yes, we do have my clicker points coming up today. So let's all concentrate and get into the Zen mode, you know. Right? So, since you already noticed that I barely have a voice, I don't want to be shouting, and I cannot concentrate if there's a lot of chit chat in the background. I have a very short attention span. If I lose my voice, I'll make sure to use my previous recording to make sure that you have everything that you need to. So before I go into today's lectures, do we have questions or comments? Yeah. Oh, put the mic up. Okay, is it better now? Now maybe, is it louder? Yeah. I don't like to talk with this thing, but I feel bad about contaminating everything. Considering my aunt just passed away because of influenza infection. Yeah, she was just fine. She got influenza. She went to the hospital. Within 18 hours, she passed away. So, yeah, I feel really bad if I contaminate everybody here with the mic. All right. Is it better now? Can you guys hear me? Okay, so please let's stay quiet, everybody. No chit chat in the background. Unless you have a question, raise your hand. Anybody has any more comments or questions or anything? Maybe the song will work too. All right, so here we go. This idea of how proteins evolved naturally, we have seen before, right? So we know that proteins are following natural laws and natural genetics selection. And basically there are this certain set of rules that proteins need to follow in order for them to evolve and be kept and be genetically selected. So proteins will evolve if the end product has the lowest possible energy state. Right? So if you make something and you're using a lot of energy and you do not achieve a lowest energy state, that means this product is not useful. But proteins, they must do something. They must have a function. Okay? So you're not just going to make some protein because you can. They must be useful. And that will be a question like, how does the system know if a protein is not useful? Don't ask me, I have no idea. But it must be useful, okay? All right. Then, this protein, it has to do the same thing over and over. It cannot decide to do something now and then tomorrow decide to do something else. No, it has to have the same function over and over. Otherwise, it is not useful or predictable. And there will be another question, how would you know if a protein wouldn't have the same function? No, it has the same function over and over. All right. The thing that it's doing has to help the organism that is making it. Because if it is a protein that is harmful for the organism, this protein is going to kill the organism. So naturally speaking, the genetics is going to be destroyed, right? Because it's not going to be naturally selected to survive. So the protein must be beneficial for the thing that produced it. So the combination of all these things is what really selects a protein for survival. Okay, so you can imagine it's also difficult to be matching every requirement for a useful protein. So here I have this image of proteins that are used for a structure. In this case, is a structure of either a planar type of sheet, or it could be a structure that makes up some tubes in case of tubulin, like makes tubules, or any other type of structural arrangement in case of, for example, viruses. So anybody here took virology? Yeah, 1, 2, 3 people. You know that viruses in proteins, viruses have proteins that do the self-assembly, right? And they just interact. Okay, guys, I hear a lot of chit-chat in the background. I will need you to be quiet because I cannot really talk very loudly, okay? So when proteins do this self-assembly, they are able to interact with each other and have their function like that, their structural function like that, as in case of tubules and in case of, as I mentioned, virus capsids. So here's a picture of a virus capsid, and you see that these proteins, like in this case, this is one type of protein that it does this self-assembly, and it's able to make this capsid just naturally, just because assembling the capsid will put the whole thing in a lower energy state. And you can also imagine this assembly is all based on electrostatic interaction, right? It's not crazy glue and it's not covalent bond. The interactions are not covalent bond. Why? Well, if it was covalent bond, by the time this virus infects a cell and is supposed to dump the genetic material inside of the cell, if it was covalent bond holding all these proteins together, the virus wouldn't be able to open up and dump the genetic material inside of the cell. Okay? So this assembly of the capsid, it has to be natural, and it has to happen if the environment surrounding the virus changes, in case of going from extracellular fluid or interstitial fluid and into the inside of the cell. The environment changes. Okay? So that is enough to open up or break down these electrostatic interactions. So no crazy glue. And here we come to this idea that you have certain proteins that can misfold and form these polymers, and the polymers themselves can act in a very strange form. These are called prions, and what happens is that the prion can infect other cells and behave as if it was a virus, or actually it is an infectious particle, okay? But this thing is only made up of protein. It does not have genetic material, okay? It replicates by bumping into other proteins that were in a different conformation, in a different structural conformation. So how is it that these prions work? Okay. So here, when a prion inside of a cell goes from a naturally folded form to an alternative folded form, then it can start to create trouble if the alternative folded form is capable of polymerizing. Okay? So what happens here? In our bodies, we have certain proteins, like for example, PRP protein in the central nervous system, that is there for a reason. We're not going to go into the reasons why this protein is there, but it's helpful, it's functional, right? It works for maintaining proper brain function. But sometimes a few amino acids in this folded protein, a few of those amino acids, they can shift the conformation from alpha helical to beta sheet. And when that happens, this protein here gets this extra property of polymerization. Okay. And how is this going to happen? Well, the first one has to be shifted naturally. Okay. So there are cases in which you have a genetic predisposition in causing this to happen. Okay. But then this shift from natural to beta sheet, this distribution, it has an equilibrium. It can go forward or it can go backwards. So this one here, if it is not polymerized, so one protein, It can go this way or it can go that way. It can go forward, back and forth, okay? What happens when it gets to this state? If it bumps into another one of these proteins that is folded properly, then this flat one causes the transition of the properly folded protein into beta sheet as well. So just this bad guy bumping into somebody who is properly folded, it will cause the shift. Okay? So now you not only you cause the shift, but you cause the initial polymerization of the protein. So now you end up with plaques that accumulate on top of each other. And this is happening inside of the cells. When you have a clump of proteins that are accumulating like this on top of each other, it's not good for the cell. And when the cell is a neuron, it's going to cause the death of the cell because that plaque is going to keep on increasing and increasing and increasing. It's going to take over the size of the cell. It's going to punch through the membrane of the cell and it's going to kill the cell. Okay? So this is a very, very big problem when those cells are your neurons. So this is the idea of prions. And again, prions are little proteins, okay? Your immune system does not detect prions. Well, this is not immunology class, but if you did, who took immunology here? 1, 2, 3, 4. Okay, who took immunology with me? 1, 2, same people, 3, 4. Basically the same people. Okay, so if you have a good idea of immunology, you know that antibodies, which are also proteins, they work outside of cells. They do not work inside of cells. Okay, and these things are happening inside of cells. The natural detector molecules or pattern recognition receptors that are inside of cells, they also are not detecting this formation of prions. Okay, they cannot take care of the prions. And there is this whole problem of how you're going to get rid of it. Well, basically, you can't. You cannot get rid of prions. They are small enough to go undetected. When they get big enough to allow some kind of detection, it's too late. Okay? So prion disease is fatal. They are all fatal. Okay? So what happens here? You have this conversion of the protein that is not toxic to another form that is basically beta strand, and it becomes toxic. Those beta strands start to make those plaques, and they're also called amyloid polymers. Okay. Have you heard of amyloid polymers in other diseases? Alzheimer's, Parkinson's, Kuru disease. Who heard of Kuru before? One person. Kuru disease. Only one person. Okay, how do you get Kuru?
Is that the one
Yes, yes. Exactly. The Kuru you get by cannibalism practices. So you guys are pretty much safe, I hope. Right? So there were some tribes. There were some tribes in Africa that performed cannibalism out of respect to the deceased. And what they did, yeah, somebody passed away, they would cook up the meat and eat it. And they had the ranking of who would eat what part. And in many societies, there were differences in ranking of citizenship in the society. So the man would get the yummy parts of the deceased. The women and the children would get the not so yummy parts, like brains and organs and things of that kind. So what happened in those cannibalistic societies, the women and the children would end up with this disease that would be very, very similar to Parkinson's. They would start developing tremors and problem with gait, with walking, and their brains would basically be destroyed. And after a lot of investigation, it was found that these prions, they were transferred from the brains of the deceased to the nervous system of the people who would consume that meat, the organ. Okay, If you want to know more about prion, I would say go on YouTube and find out more about it. It is fascinating. At least I think it is fascinating. Another similar disease is CJD. So this is the Creutzfeldt-Jakob disease. Okay. So you have two forms of CJD. You have familial form and you have acquired form. The acquired form is mad cow disease. I hope everybody heard of mad cow disease over here. Okay, who has never heard of mad cow disease? Okay, well, one person. You never heard mad cow, like 1992, England issues? No. All right. Make sure you look on YouTube about mad cow disease. Okay. So what happened is that even today, you have cases of mad cow disease, even nowadays. Why? Well, because in this case, the cattle, okay, the cows, they were fed contaminated organ source of animals that had prion diseases. So the cows, just like the cannibalist society, the cows acquired these prions, it went through the intestine, it went to the central nervous system, and they developed prion disease, central nervous system prion disease. So that is the mad cow disease. It is prion based. Yes. Well, you have the PRP protein that is not considered a prion. So if it is not a prion, then it's not infectious.
So there's no like prion?
No, the prion is by definition the thing that causes an infection. Yeah. The, Okay, yeah, this one, good point. Here, where it says normal prion, it should be normal prion precursor. Okay, it should be a precursor protein. So it's not the prion itself. So you have a normal protein, it should say normal protein. And then diseased or prion conformation. Yeah, so if you say prion, that is something that causes disease. Okay? This should be normal PRP protein. So if it becomes a prion, this is when you have infectious protein that is capable not only of infecting nearby cells, but is capable of crossing species. So if you eat a hamburger that has prions, you have a chance of acquiring prion disease, or in that case, mad cow disease. Okay. Now, how do you destroy prions? Yeah.
Not with the vaccination.
No, there are no vaccines against prions as far as I know. Yeah. Huh? Heat, that's the only thing. Not only heat, but it has to be incineration. You have to burn the thing to carbon dioxide. Yeah. Doesn't matter. Yeah. If you're cooking a hamburger, you're going to kill bacteria or viruses, so that you're not going to be contaminated with certain bacteria or viruses, like bacteria like E. coli, that there are some very bad strains, right? But if you're trying to get rid of prions by cooking, forget it. You can put this thing in autoclave and it's not going to destroy it. You can try to use bleach or alkaline type of reagents or cleaning products or disinfectant. You can't get rid of prions. The only way to get rid of prions is by incineration, making ashes of whatever it was. If you're going to do that to your hamburger, you can't eat it afterwards, right? It's like ashes, right? So, yeah, no, well-cooked hamburger, that's not going to do it. Okay. And then there will be the other question. Didn't I just mention that you have two forms of prion disease, like genetically inherited, and then the mad cow disease, right? What do you do with genetically inherited diseases? First of all, you can only treat the symptoms, okay? You cannot get rid of the disease. If you have genetically inherited prion disease, it's a ticking bomb, you're gonna die. The good news is that we're all gonna die, sooner or later. But anyway, at least you know why you're gonna die, okay? All right. Now, how do you get rid of, let's put a situation, you have somebody who is undergoing brain surgery, thinking that they have Parkinson's, for example, right? Ends up the person has prion disease, and you just found out it was prion disease after some biopsy sample of the brain, right? So now what happened? You have the surgery equipment all contaminated with prions, right? You have those very expensive stuff that goes into people's brains. Those are all contaminated with prions. Can you use that again in another patient? No, not going to happen. Unless you incinerate it. Yes, but not for prions. Yeah, question was, can't you just use heat to sterilize equipment? Yeah, but it doesn't work with prions. The whole thing. Actually, I learned last year that the military has some form of sequential high water pressure cleaning system to try to salvage the equipment that got contaminated with prions. Would I trust it? I don't know. Okay, but it's a big problem. It's a big problem. Okay. So that was the history with the prion. So here, causes of, okay guys, I still hear chit chat in the background. I know everybody's very excited about your brain becoming a sponge. So let's focus here, there's more good stuff. Okay? So causes of prion. What happens at first, you had this conformation change in which a huge part of the protein had alpha helices, and then naturally or not so naturally, you get a lot of it transforming into beta sheets. The prion is going to serve as a template or as a catalyst. What do you know about proteins being catalysts? Most catalysts in your body are proteins, and so is this guy. Okay, it's a protein. So it serves as a catalyst, and it serves as a platform to transform the good behaving, well-behaving guys into bad guys as well. Okay, so once you start this polymerization process, as far as I know, so far, you can't stop it. All right. All right, then you get the plaques, and then by the time you look at an autopsy of a brain that diseased from prion disease, you're going to see that it looks like a sponge. It's full of holes. Okay. It's called spongiform encephalitis. That's another name for it. And here comes your first question. And here I shall open this. The class should be on. Okay, and it started to count up. All right, prion disease are best classified as A, infectious protein folding disorders, B, viral encephalitis, C, autoimmune disease, D, genetic bacterial infections. So you guys have a few more seconds because this thing is counting very quickly. Put in your votes. Oh, it's not stopping. Does anybody need more time? Yeah? Yeah. I think it's going to count up to 1 minute. So you have like 20 more seconds, 15 more seconds. All right, make sure you cast in your votes. Okay. So everybody voted, I hope. Anybody need more time? No? All right, what's your answer? It is A. All right, I had a few people who answered B, C, or D, mainly because I hope it was an error pressing the button. Okay, we just talked about this. Okay, so now this thing will not go away. All right. All right, so now let's get to the part of protein function. Right? Protein function. Functions of proteins. So this thing is really easy because I have brushed on it before. Right? This thing, this part. You have a certain shape on the surface of the protein. Remember, when proteins are folding, they are doing so at the same time that they are being produced. So you have that co-translational folding. And does it use energy for the protein to fold when it's coming out of the ribosome? No, right? No energy needed because you have those interactions that occur naturally. And remember those van der Waals interactions. You remember the phi and psi angles, right? And all that stuff. So it has certain chemistry that allow a certain type of protein interaction. Now, all proteins, they have to do something, and they are able to interact with other molecules. Okay, all proteins are going to interact with something. Okay? A lot of times they're other proteins, sometimes they're substrates, and sometimes they are things that are other than proteins like DNA and RNA and etc. Okay? The binding of the protein to the something it works with, these are all electrostatic interactions. Okay? Now, after the initial electrostatic interaction, you can have some temporary covalent bonds that will form between the protein and the substrate. And that involves the formation of intermediates of this process, okay, of the enzymatic process. Those covalent bonds are temporary, they're going to form and they're going to break. It's part of transforming your substrate into products. And we're going to get into more mechanism of that later on. But at the end of the catalysis, you end up with the protein in the same shape that it was before it started. So proteins are not consumed by the reaction. Okay. The binding of the protein to whatever it's working with is really, really specific. Okay, so high specificity. That means high affinity of the protein to the substrate. Okay. Now, if you took other classes like biochemistry or other biology classes before, you know that there are a couple of theories of protein interaction. You have like the lock and key, and you have the induced fit model. Right? Does it sound familiar? Induced fit, lock and key. The lock and key is kind of primitive because it has this very rigid form of interaction. Induced fit is more like, I assume that is how it works, because you have constant small modifications of the shape as you're getting from reactants to intermediate to products. All right, so here is this idea of complementarity. So the complementarity of the protein with the substrate and the way that they interact by electrostatic forces. So this is going to be, although we see in 2D here, this is a 3D shape that is making use of those static interactions. All right, so the thing that is going to bind to the protein is going to bind at a site that is specific for it. So the site is called the binding site. The binding site is specific for the substrate. Okay? The enzyme itself might have other sites, but the site in which you're going to have the chemical reaction is the substrate. So here you have an idea of how the interaction is going to happen. In case of a protein that is, let's say, a globular protein that is going to interact with a substrate, and the way that the substrate will fit in this pocket that is highly specific. So here is showing that, like a puzzle piece, you have certain shape that will allow this shape of protein interaction. But keep in mind that this stuff is all globular. When you look at protein shape, you're looking at this 3D blob. Okay, a lot of time blobs look the same, but the charges around the blobs are different. The sequence of amino acids, the sequence is different between different blobs. Okay, all blobs look the same. So even if another blob comes in here and is able to fit, it will not have the required electrostatic interactions to interact with that protein. Okay, so electrostatic interaction at the site meaning here is active site that is inside of the protein. Mainly, you don't have a lot of water molecules inside of this thing, okay? The interaction, it has to be because of those side groups that are present in your substrate. So this can be inside the cavity of your enzyme, or it could be on the surface of this protein, and this would be basically all exposed to the watery environment, right, to the aqueous environment. But now here, this interacting surface is going to have specific properties that will interact with electrostatic charge, in this case of a DNA molecule, right? And this is more like, it is intended to work in a watery environment, in comparison to this one here, that is intended to work in a hydrophobic environment, okay? Because inside the protein pockets, you have very, very little water. All right, so binding of proteins. When you bind a molecule to your enzyme, the enzyme is going to change the folding of the molecule, and the molecule is also going to influence the shape of that enzyme. Okay, so you're changing the protein environment, and you're changing the protein shape. Okay, so that is really what's going to lead to the interaction, to the protein function. All right, so when you say changing shape, it means conformational change. Okay? So let's see more about this. Okay, so let's see. When you change the shape of the protein, you're going to activate certain behavior that was not really active before. Okay? You can make new binding sites, you can change other binding sites, you can gain or lose function. Right? Now let's see here. As I mentioned, the inside of the protein is going to work because of certain electrostatic interactions. So here you see, in the case of a unfolded protein, when it folds to its proper shape, you're going to have those side groups that are going to guide the function of your active site. And again, the specific pocket is going to interact with one particular type of molecule. And in this case here, it is showing the interaction of cyclic AMP, which is this molecule right here. It is showing the interaction of cyclic AMP at this pocket of this protein. So when you look at the possible interactions here, you're going to see hydrogen bonds. Okay? You're going to see electrostatic interaction, positive and negative charge. These would be like more like ionic interactions, right? You see more hydrogen bonds. So everything here is not covalently attached to the protein. Okay? Now, if you're wondering how this thing is a cyclic AMP molecule, so I added this other little figure here, this diagram over here. So you should recognize this as being some kind of a nucleotide, right? That was in your test, right? What do you need for a nucleotide? You need a little house, that is your sugar molecule. You need a base, that's your little garage, right? This nitrogenous base. And you need a phosphate pool. So in this case, the guy only has one pool, like phosphate. So this is your AMP molecule. And you also recognize this is the oxyribonucleotide, right? Do you know the difference between the oxyribonucleotide and ribonucleotide? Yeah, the ribonucleotide has another hydroxyl group here, OH, right? And I didn't get to do my little walk dance for the class, how to distinguish the oxynucleotide, deoxyribose from ribose, right? You know what I'm talking about? No, okay, so this guy here is deoxyribose. If it had two hydroxyl groups, it would be ribose. If you don't remember which one is which, you think about this little guy being a little man walking. So the top here, the O, it would be the head of the person. And in case it's missing a foot, it's going to walk like deoxyribose. And when you have a figure, ribose. So that's my ribose walk. So if you look back and you see Missing a foot is deoxyribose. All right. And here you see that this phosphate group is happy in its pool state, right? You put a little circle here, you see a pool, and you can jump on it. Okay. Now, comparing it to this guy here that has a cycling, it has a circle made up of the phosphate. Okay, it's a cyclic thing out of there. Okay, it's some kind of a phosphodiester bond going on over here. Okay, what happens here? This structure is not very stable. All right, this structure can be broken down easily by this other enzyme, which is represented over here, okay? Because this shape here is going to fit perfectly well in this little pocket. Now, you look at this molecule that is inside of the enzyme, and you're going to see here is your base, here's your nitrogenous base. This guy here is your deoxyribose, but how is that happening? Look, this deoxyribose here, it has a kind of a joint with the phosphate, okay? So it looks like if you break the bond over here, you're going to have your ribose out of there, but that's not what's going to happen, okay? But you see here, you have a lot of these negative charges that are on this side of the molecule. And when you look at this ring over here, you see that the phosphate is part of that ring. Okay? So this is why you can identify this blob over here, that it is a cyclic AMP molecule. Okay? Why am I talking about cyclic AMP here? Well, it's a very, very important second messenger. Okay? Important second messenger. We're going to see more about that later. But keep that in the back of your mind. Since it is an important second messenger, it cannot last forever in your system or in your cells. Otherwise, it's going to keep on giving messages. It's a messenger. It does something to the cell. It's going to keep on giving messages and cause some cell stuff to happen. Okay? That means you're going to have enzymes to break cyclic AMP into what? When you break cyclic AMP, you get what? Well, what you're going to break out of the cyclic AMP? That would be the first question. You break this cyclic structure. So when you break cyclic AMP, you end up with AMP. So AMP, it's not a very good second messenger. It does not do the job the cyclic AMP can do. Okay? Now keep that in the back of your mind. We're going to learn more about that later. But yeah, the enzyme here is very specific to accommodate cyclic AMP on its active site. All right, so more functions of proteins. We looked at prions. That's actually a very bad function of proteins. It's a disease kind of thing. And we looked at conformation of protein with a specific active site. Okay, so now we're going to look at all these other functions, which we have also briefly mentioned before, but now we're going to go into more specific types of classifications. Now, so first one, this is one of my favorite proteins. Those are antibodies. I don't know if I like them as much now as I used to before 4 months ago. Anyway, long story. So, did I ever tell you why I got stuck in Brazil? No, okay. So let me pause this, since we have not chatted for quite some time. So I'm gonna have the look. Did this thing stop? Okay, now. Okay. So antibodies, right? So what happened here? So I had to draw her blood and get antibodies titers, antibodies against rabies, right? There is a certain level of antibodies that the CDC requires in order for the dog to fly from a, what else? High risk rabies country, which Brazil happens to fall into that category, to the US. The antibody level is like 0.5 units per milliliter, something like that. Poor little Lola had 4.0, nearly 10 times the amount of antibodies against rabies. Okay? And again, they did not let me fly. Why? Why? Because the CDC requires 28 days after drawing the freaking blood. Why? Why? Don't ask me why, it doesn't make any sense. But I had to be sitting there and waiting, okay? And this whole idea of the microchip itself. The microchip has to be implanted after the antibody, the vaccine booster, otherwise the vaccine is not valid. Who made this rule? For goodness sake, it has to be some politicians that never took immunology class in their lives, or never had a friend who understood enough from immunology to tell them that that doesn't make any sense, right? I called the CDC so many times. I called the FDA so many times, and guess what? You heard about this current administration laying off people that work in this kind of government agencies? They managed to lay off everybody with a thinking brain out of those agencies. I swear, if I was talking to artificial intelligence, at least there would be intelligence in the wording. There was this one person, Miss Becky, is the only person that deals with importation of dogs. One person. Every time I would call there, she would read to me out loud 3 pages of instructions of CDC requirements for importation of dogs. Okay, I'm traumatized. Anyway, didn't happen. So that's why I couldn't come back home. But finally, and then yesterday, oh, if I start telling you about the nightmare yesterday, okay, let's not get there, we'll never finish the class. Okay. Anyway, antibodies. What is it that antibodies do? Right, let's get into the same mode. Yes, the question, no, it's scratching. Me too, I'm still scratching my head. All right, everybody quiet. Everybody quiet. Antibodies, they are your defense of your adaptive immune system. They will fight pathogens that your body has never seen before. Even without vaccination, the body can predict what kind of thing is going to attack you. Bye, good weekend. All right, you have antibodies for that. Most kind of diseases, you got antibodies for that. And certain diseases like rabies, right, caused by viruses, influenza. Yeah, did everybody get your flu shot this year? Oh, I'm in the wrong country. It's important in Brazil because it's winter over there. Anyway, so I cannot take influenza shot because they use attenuated virus. It means it's not killed, right? My body, when it sees an attenuated influenza virus, it goes crazy. I get the flu just by looking at the virus. So I cannot take the shot. If you took your influenza shot this year, you're probably not going to get contaminated by whatever virus I have right now. But I'll feel bad if I contaminate you, okay? But yeah, antibodies will do the work. They will attack things before it makes you sick. Function of proteins. How is it the antibodies look like? You have 4 chains of proteins, 4 polypeptides. Why is everybody leaving me? Am I blowing up the time? No, I still have like 15 minutes. Okay. Bye. Have a good weekend. So, antibodies, they are made of 4 polypeptides. What is the highest level of protein instruction you have here? Quaternary. You have more than 1 polypeptide that is interacting. You look at what keeps this polypeptide together. Those are disulfide bonds. You have interchain disulfide bonds. You have intrachain disulfide bonds. So interchain here, intrachain there, is packed with disulfide bonds. What is the name of the residue that allows for disulfide bond formation? Cysteine, exactly. Disulfide group, right? So here's the basic shape of your antibody. 2 heavy chains, 2 light chains, and it makes this little white shape that is so adorable. Other proteins. You have proteins that are transcription factors or enhancers. Either one, you have the transcription factor or enhancer is a different type of protein, okay? And those are going to interact with the DNA And they're going to coordinate what kind of genes are going to be expressed depending on the type of cell that you're dealing with. Okay? So remember that when the egg gets fertilized, you have one cell that has a full pack of DNA of chromosomes, right? This one cell starts to divide. So it goes 2, 4, 8, 16 is power of 2, right? So then you get your morula, it's a bunch of cells in there. And then you start to have differentiation. Some cells are going to be part of your ectoderm, some other cells are going to be part of your mesoderm, some other cells are going to make the endoderm. And then you have further differentiation, right? Now, when the cell is differentiating, the cell starts to express certain transcription factors. If the cell has a certain transcription factor, then you're going to be making those enhancer proteins or other transcription factors to further differentiate and keep the cell at a differentiation level that is useful. Okay? So you look at a cell in your liver is different than a cell in your brain. So all your brain cells right now should be activated by NMDA receptors that would tell you to keep quiet when I'm talking. All right? So cells are expressing different transcription factors. Differentiation of transcription factors will tell you what kind of cells you're going to end up with. Okay? Very, very important for regulation of gene expression, which of course we're going to be talking about more later. Then you have these kinds of proteins. There is transport of molecules. How is it that you get molecules transported, for example, from your gut to the rest of your body or to your liver? If you have a meal that is composed of that yummy hamburger, hopefully without prions, right? The hamburger is going to have fat, right? And the fat, yeah, depending how much fat is good fat or bad fat, there's some fats that, especially the unsaturated fats, they're actually good for you, right? You want to transport that from your gut to your liver, but fat is water insoluble, so you need some protein for that transport. So here is an example of a kilomicron that is made up of proteins and a piece of membrane, okay, that you have your phospholipids, right? Is made of proteins and lipids in a way that you can pocket those fats and transport them in a water environment from your gut to your liver. Right? So this is a kind of transport molecule that we're talking about. Proteins are important for that. Another type of transport molecule is myoglobin and hemoglobin. And those are going to transport oxygen as well as carbon dioxide, but oxygen especially. Oh, here we go, oxygen transport. Okay, you have here one peptide, polypeptide chain, that's your myoglobin. You have here 4 polypeptide chains that get together to form your hemoglobin. Both myoglobin and hemoglobin will transport oxygen. Now, myoglobin really likes to hold on to that oxygen. Hemoglobin, when it gets close to myoglobin that doesn't have oxygen, hemoglobin donates the oxygen to myoglobin. So they're both transporting oxygen, and you're going to have them in different tissues depending on what their functions are. Both hemoglobin and myoglobin have in their core this iron center is heme, okay, heme iron center that directly binds to oxygen, okay? And then we're going to go, when you get into the hemoglobin, I can tell you some horror stories. All right. We have some proteins that are very important for gradient production. So you have in this case the ATPase, right? You have this sodium potassium ATPase, and we have seen that before, right? 3 sodiums out, 2 potassium in, and you are using phosphate for that. So enzyme that is using phosphate is ATP synthetase in this case. Okay. We're gonna have more on these stories. So enzymes for transport, and then you have some structural proteins, and those are for maintenance of cell structure as well as of body structure. You have some examples here of structural proteins, especially collagen. That is really the most abundant structural protein in your body. Okay, where do you find collagen? You find them in tendons, skin, in the cornea, in cartilage, bone, blood vessel, gut, basically everywhere in the body you find some kind of collagen. And also extracellular matrix is very well known for having collagen. And you can imagine you have different types of collagen. The type that is like type 1 is the strongest, is the most resistant type of collagen. And as you go up in number, like type 10, 11, 12, those are very much soft type of collagen protein. Okay. How do you make collagen in the cell? Well, it's a protein. So it is made in the plasmid reticulum, well, because it's meant to be deposited outside of the body, right? So it goes through the rough ER, and then there's some other processes. Now, we're going to get into these aspects of collagen later. Now, you should know that the proteins that are making collagen, they need a specific molecule that makes them work better. That molecule is going to bind to a site that is not the active site. So we call that the allosteric site. The molecule we're talking about is vitamin C. We're going to find out more about allosteric sites and binding interactions. Right? If you do not have vitamin C, your protein enzyme cannot make collagen very well. Okay, you cannot do some modification to the collagen to make it stronger. So without vitamin C, you cannot really build a strong collagen. That means all of the tissues in your body that are depending on this collagen building up, you're going to have some deficiencies. Okay, so if a person is vitamin C deficient, you're going to have problem with collagen production because you're not going to be able to cross-link those helices, those alpha helices of collagen. Then what happens? You end up with a disease called scurvy. Anybody heard of scurvy? Yeah, that's the pirate's disease, right? Scurvy, and you think about the face of the pirate, sunken eyes, pale skin, teeth missing, broken legs, missing hands. I don't know about the missing hand, but yeah, they can break bones easily. Right? So scurvy is a typical disease for lack of vitamin C in the diet. So here you see the treatment of a pirate that is suffering from scurvy. What was the guy doing? Yeah, I think it was actually lemon. Yeah, the guy is stuffing this poor pirate's mouth with a lemon. No, it's just giving him lemon juice, I hope. Stuff the poor. That's it. So you give vitamin C and you treat and you cure scurvy. You don't really see nowadays scurvy cases, except for disease states, other diseases. So here, other structural protein that is quite important, elastin, also very, very abundant in your body. You're going to find that in the skin. You find that in your lungs. Why is elastin important for your lungs? Well, because elastin can stretch out and recoil, just like a rubber band. Stretch out and recoil. Your lungs are stretching out and recoiling all the time. If it doesn't, you die. Right? So elastin is very, very important for this flexibility of tissues. It's also very important for some ligaments and other things. Okay? Skin, elastin in the skin. You can stretch and it will recoil. If it doesn't recoil, you are dehydrated. Don't start pulling everybody's skin just if you're dehydrated. Just drink some water. All right, then you have the synthases. What is a synthase? Synthase takes advantage of electrochemical gradient and makes ATP with that gradient when the protons go through a little tunnel inside of that protein, inside of the enzyme. Okay, so that's your ATP synthase. It gets energy from that proton gradient, and it's going to make ATP. Okay, and And then you have other enzymes that are called the synthetases. Okay. It uses ATP to do something else. So you know the difference between a synthase and a synthetase. I always get confused, that's why I put this little chart in here, so it's FYI. You can read about that on your own. Now that I do not provide the class, okay, so you have like 4 minutes. Okay, you read that on your own. And then you have these motor proteins. Those are really, really cool. Right? They are very, very important for muscle contractions, which is not what I'm showing here. And they are very important for crawling of cells after some bacteria and in case of those immune cells, right? They're very important for cell division, lines up the chromosome in the middle of the cell, and they're extremely important for vesicular transport inside of the cell. So these proteins will literally like walk on a little track, and they use ATP to change their conformation and really put like one foot in front of the other foot and walk in those little tracks. And I believe I have here a little movie showing this dyning protein that is walking on a tubulin track. And of course, if the thing is walking, it has to be carrying something, so it can be useful. So you're gonna see here, this little dyning is carrying a little vesicle. I mean, it looks huge in comparison to this little guy here. So let's see here if this movie is going to work. Yeah, look at that, look at that. Look at the little feet. That is so neat. And this guy is carrying the whole world on its hands. Look at that. It's a whole, forget about the little ants carrying a leaf. This guy is much more impressive. Look at this. And he's bending his back and doing this whole work. Yeah, I love that movie. So those are your motor proteins. Okay, and another question just before we go. Let's read it here. I'll start the question. Okay, antibodies. Antibodies are a sample of proteins that function in what? All right, cast your votes before everybody runs away. Right, get like 5 more seconds. Stop here. Does anybody need more time? No? Everybody's done? I'm gonna stop it. Okay. What's your answer? B, immune function. I still have some people answer something else. Make sure you understand what antibodies are doing. Okay.